Hi, my name is Taylor, and today I'll be sharing some information on tempeh, a fermented soy superfood. Tempeh originally hails from Indonesia, where the fungus Rhizopus microsporus oligosporus was harvested from the leaves of native hibiscus trees and used to initiate the fermentation of soybeans. This not only enhanced the flavor but allowed for increased shelf life and improved nutritional properties of the soybeans. The process of making tempeh is fairly straightforward and requires very few ingredients and minimal processing time. Soybeans are first cleaned, soaked, and dehulled. They are then boiled to remove any pathogenic or unwanted microorganisms and, once cool, inoculated with a mold, Rhizopus oligosporus. Once inoculated, the soybeans are incubated at a temperature of 27 to 32 degrees Celsius for 24 to 72 hours. A variety of microorganisms have been isolated from fermented tempeh. While Rhizopus oligosporus is chiefly responsible for the fermentation process, Klebsiella pneumoniae and several species of Lactobacillus are also thought to provide added benefit to gut bacteria and the human microbiota when ingested. Rhizopus oligosporus belongs to the domain of Eukarya, the kingdom of fungi, and its further taxonomic classifications include the genus Rhizopus, the species Microsporus, and the variety Oligosporus. It is a heterotroph, meaning it gets its carbon from sources other than the sun, usually dead and decaying organic matter. Rhizopus oligosporus is truly a fantastic fungi. This mold is responsible for breaking down major molecules in the soybeans into smaller metabolites, or micronutrients. Soybeans, for example, contain high numbers of proteins. Rhizopus oligosporus uses the metabolic processes of fermentation to break these macromolecules up into their smallest subunits, amino acids. These amino acids are used by our bodies to form various other functional and life-sustaining proteins. Studies on amino acid concentrations during the fermentation of soybeans have measured levels of each of the 20 amino acids at 0, 24, and 72 hours after inoculation with Rhizopus oligosporus. In the graph, you'll see data from the 9 essential amino acids, showing that across the board, levels of amino acids increased from the time of inoculation to the 72-hour mark. As you can see, some amino acids increased more than others. Lysine, for example, increased by nearly 4,000%. Rhizopus oligosporus is just one of many microorganisms that, through its own metabolic processes, adds nutritional benefits to foods within the animal diet. It demonstrates that we not only benefit from, but function best in symbiosis with the microbial community, thanks to processes that are mostly invisible to the naked eye. Thank you.
My name is Kiana, and my chosen microorganism is Aspergillus oryzae, and I will talk about how it's involved in soy sauce production. Just to begin, I'd like to give an overview. I will summarize the food process, and then I'll get into the brewing and metabolic process and how my microorganism plays an important role in fermentation. So, soy sauce, we all know it. It's a flavor enhancer in cooking, and it is a general table condiment. Ingredients used in soy sauce production include soybeans, salt, wheat, water, lactic acid bacteria, and fungi. So, you're probably wondering, how does all of this turn into soy sauce in the end? So let's get into the brewing and metabolic process. It includes koji fermentation and moromi fermentation. And we're mainly gonna focus on koji fermentation, as this is where my organism plays an important role. So again, Aspergillus oryzae, you're probably wondering, why is it moldy? Why do we put that in soy sauce? I thought the same thing too, but after further research, mold is a type of fungi, and it plays an important role in breaking down macromolecules. And without getting into too much detail about it, it's essentially a filamentous mold that thrives in aerobic conditions, otherwise known as it's an obligate aerobe. It requires oxygen to grow and does not generally grow under anaerobic conditions. So, in this first stage, it produces enzymes like protease and amylase, and these enzymes are responsible for hydrolyzing proteins into amino acids and starches into sugars. And we're in the stage for about 48 hours to break down all these macromolecules. And then we'll move into the second stage, moromi fermentation, which is essentially koji fermentation put into a brine solution. And it sits in this brine solution for 6 months to a couple years to continue the fermentation process by using lactic acid bacteria and yeast to further metabolize these products. And then by the end of both stages, the mold is no longer active, but its enzymatic byproducts remain, and it enhances the overall taste. That is essentially soy sauce production. I hope you enjoyed it, and thank you for listening.
Every day we live and every meal we eat, we influence the great microbial organ inside of us, for better or for worse. Kombucha is a fermented tea that is a probiotic, which contain live microorganisms known to increase the diversity of the microbiome in our gut. It has been used for thousands of years, dating back to 220 BC. Kombucha originated in China for its healing properties. Kombucha is made by introducing a SCOBY, symbiotic colony of bacteria and yeast, into brewed black tea with sugar. The tea sits and ferments for 1 to 4 weeks and is ready for consumption. When bottled, carbon dioxide gets trapped and makes it bubbly. Kombucha is made up of 200 plus microorganisms. One of the predominant microorganisms is Acetobacteriaceae. It is an AAB, acetic acid bacteria, found commonly in fermented foods. This is what produces the vinegar flavor. Another common microorganism in fungus is Zygosaccharomyces. The Acetobacteriaceae family is a gram-negative rod-shaped bacterium that oxidizes sugars and produces acetic acid. Acetic acid bacteria dominate the bacterial community of kombucha and drive the fermentation process. Some of the AAB is produced through carbohydrate metabolism, but most of it is through the oxidation of ethanol. Kombucha has grown to be increasingly popular over the last few decades. You can now buy kits to begin the tea fermentation process in your own kitchen. These kits include a starter tea to inoculate your new tea. Once your kombucha has had time to brew, it should be moved into the refrigerator to slow down the fermentation process. Kombucha is alive with yeast and bacteria. Even after stored in the fridge, it will continue to ferment, but at a slower pace. These live cultures are what make kombucha full of probiotics and increase our gut microbiome and boost our immunity.
Tuber melanosporum. These mushrooms are some of the most sought after and exotic foods in the world. They are native to France, but our climate here makes it ideal for harvesting, so we are able to forage them here in Oregon and Washington as well. Mostly in forests and the coastal range foothills. Truffles are not only difficult to forage, but even more difficult to cultivate. Tuber melanosporum contains several antioxidants, so they have many potential health benefits as well. They are considered rare, highly seasonal, and a luxury because they are expensive, costing up to $4,000 per pound. But they sure do taste good.
Hello everyone, today I'll be walking you through the fascinating world of beer brewing, focusing on the essential role played by microorganisms, particularly the yeast Saccharomyces cerevisiae. Beer brewing is one of the oldest and most widely practiced forms of food production. In fact, beer has been enjoyed for over 7,000 years, with its roots going back to ancient Mesopotamia. Today it remains one of the world's most consumed beverages, evolving into both big business and a popular home brewing hobby. The key ingredients of beer, malted barley, hops, water, and yeast, come from various sources around the world. Barley is mainly grown in Europe and Northern America, providing fermentable sugars. Hops, which are added for flavor and bitterness, are often sourced from regions like Germany and the Pacific Northwest. Saccharomyces cerevisiae, or brewer's yeast, is the star of the fermentation process. This yeast converts the sugars from barley into alcohol and carbon dioxide, giving beer its alcohol content and characteristic fizz. It also produces byproducts like esters that enhance the beer's flavor. The fermentation process is where the magic happens. Once the malted barley is mashed into a sugar-rich liquid called wort, yeast is added. The yeast ferments the sugars, producing ethanol and carbon dioxide. This process also generates secondary metabolites that contribute to the aroma and taste of the beer. In large-scale commercial brewing, S. cerevisiae is carefully cultured from pure strains to ensure consistency, while small-scale homebrewers often experiment with wild strains to develop unique flavor profiles. The yeast plays a vital role in both settings, with commercial practices focusing on efficiency and uniformity, and homebrewing offering more creative expression. After fermentation, most of the yeast cells die off or become inactive, but some remain in the final product, especially in certain bottled beers. These live yeast cells can continue fermenting any remaining sugars, enhancing the beer's flavor and carbonation over time. With modern techniques like genetic modification and selective breeding, brewers are constantly innovating yeast strains to enhance beer quality. These advances allow for improved fermentation efficiency, better resistance to spoilage, and the development of new, exciting beer flavors. Beer brewing is a beautiful blend of science and art, with yeast at the heart of it all, driving the fermentation process that gives beer its unique qualities. Thank you.
My presentation is on the making of chocolate, my favorite snack, and the process of using a specific yeast called Saccharomyces cerevisiae. The word chocolate comes from the Aztec word xocolotl. Chocolate has been around as a commodity since 1500 BCE. It most likely got its start being used for ceremonial purposes by the Olmecs in Mesoamerica. It has also been used in cooking, baking, and candy, as we all know, but also as an aphrodisiac. Chocolate comes from the tree Theobroma cacao, more commonly known as cacao trees. Cacao pods are large and can be up to 14 inches long by 5 inches wide. The beans of the cacao pod are fermented, then dried, cleaned, roasted, separated from the shell, milled into chocolate liquor, hydraulically pressed, filtered and separated into cacao powder and cacao butter. Then these raw ingredients can be mixed with sugar and other ingredients formed, packaged, and sold. Saccharomyces cerevisiae, commonly known as baker's yeast or brewer's yeast, is the most common yeast used in the primary fermentation of cacao beans. It is a eukaryotic organism, facultative anaerobe, with shapes ranging from spherical to ellipsoid, depending on whether they are in haploid or diploid phase. They can reproduce by both sexual and asexual reproduction. Asexual reproduction is the most common method, and a process called budding produces genetically identical cells to the parent cell. The photo shows some buddings on parent cells. The metabolic process involved in making chocolate involves fermentation, including anaerobic fermentation and aerobic fermentation, but Saccharomyces cerevisiae is responsible only for anaerobic fermentation. Other microorganisms, even wild ones, including other yeasts and bacteria carried by workers, flies, and the environment, also help with fermentation. All of these organisms contribute to the distinct flavors of chocolate. If you're looking at the photos above and wondering if the flies end up in the chocolate, the answer is yes. Unfortunately, according to the FDA, it's okay if there are less than 60 or more insect fragments per 100 grams of chocolate. In primary fermentation, the S. cerevisiae converts sucrose, glucose, and fructose via glycolysis, producing 2-ATP and pyruvate, which ultimately yields carbon dioxide and ethanol. During primary fermentation, a co-occurring fermentation of bacteria is also happening, which also produces lactic acid. The final products of primary fermentation are sweatings and pulp, shown in the top photo, CO2, ethanol, and lactic acid. These products go into secondary fermentation that is done primarily by bacteria, which I will not go into details about. After secondary fermentation, the yeast and bacteria die off, and the product is now chocolate liquor, which goes on to become chocolate, as described earlier in the presentation. And that is how Saccharomyces cerevisiae is involved in the production of the delicious, joyous chocolate that so many people love, insect parts and all.
For this assignment, I'll be looking at Bacillus subtilis variationata and fermentation of food. So fermentation is an aerobic process in which microorganisms like yeast and bacteria break down food components into other products, which gives fermented foods their unique and desirable taste, aroma, texture, and appearance. And popular examples include kimchi and alcohol, and various microorganisms are used in fermentation, but certain foods use certain microorganisms. And there is an increasing popularity of fermented foods because of their gut and GI benefits and a long shelf life. So natto is a Japanese side dish composing of fermented soybeans, and they're known for their sticky and stringy texture along with their pungent smell. They also have a salty and bitter taste to it, and it's usually consumed for gut health, promoting properties, and long shelf life. And the main organism used in this production process is called Bacillus subtilis variation natto. So Bacillus subtilis variation natto is a rod-shaped, aerobic, endospore-forming, gram-positive bacterium that helps keep normal balance of intestinal flora. And when added during fermentation as a starter, B. subtilis variation natto produces Y-polyglutamic acid, which gives natto its viscous texture, and the bioactive factors involved in the fermentation of natto include nattokinase, bacillopeptidase, and dipicolinic acid.
Hello everyone, my name is Tuzar, and today I'm going to talk about the amazing role of microbes in yogurt production. In yogurt making process, yogurt start with milk, which is best heated to around 85 degrees Celsius to denature the proteins. This step ensures a smooth and creamy final product. After heating the milk, it is cooled to about 43 degrees Celsius, which is the ideal temperature for the yogurt culture to thrive. And we do have key microbes in this production, which are Lactobacillus tuberculosis bulgaricus and Streptococcus thermophilus. The Lactobacillus tuberculosis bulgaricus, it is the rod-shaped gram-positive bacterium. It thrives in acidic environment created by lactic acid. And it ferments lactose, the sugar in milk, into lactic acid, which helps to thicken the yogurt and make it its tangy flavor. And the next one is Streptococcus thermophilus. It is a spherical gram-positive bacteria that forms change. It is facultative anaerobes, capable of growing in both aerobic and anaerobic conditions. It ferments lactose into lactic acid, and it works in synergy with L. bulgaricus, enhancing each other's growth and activity. This cooperation results in a balanced flavor and creamy texture in the yogurt. And we do have the fermentation process, which is lactic acid fermentation. So during fermentation, the bacteria consume lactose and produce lactic acid. This acidification causes the milk protein to coagulate, transforming the liquid milk into thick, creamy texture we associate with yogurt. This process typically takes between 4 to 12 hours. And we do have health benefit in yogurt. So the yogurt is alive. The bacteria is alive in yogurt, which is probiotics. It helps digestion, boosts immune system, and it also even alleviate lactose intolerance by breaking down lactose. And in summary, the role of microbes in yogurt production is really crucial for us. These tiny microorganisms not only enhance the taste and texture of yogurt, but also provide significant health benefit, making yogurt a nutritious and beneficial addition to our diets. That is all for my presentation. Thank you so much for listening. Have a great day.
How do we go from this sloppy mess to this? It's called the sourdough starter, an ancient symbiotic relationship among microorganisms, grain flour, and water that can be dated as far back as 3000 BC in Egypt. Contained within this sour slop is the magic of sourdough bread, a symbiotic ecosystem formed when flour and water interact with microorganisms, resulting in spontaneous fermentation in the production of unique byproducts. Fermentation is a natural process where microorganisms, like bacteria and yeast, break down sugars into other substances, often producing energy in the process. Starters are inoculated by microorganisms from various sources, including air, the baker's hands, and even the flour itself. The products of fermentation, while considered waste to the cells, can actually be useful for humans, and in this case, sourdough bread's characteristic texture and flavor are due to the ethanol, acids, and carbon dioxide produced by them. Among the countless microorganisms found in starters, Lactobacillus sanfrancesensis, a lactic acid bacteria, has consistently been identified as a dominant species in sourdough starters studied by scientists. The name refers to the city of San Francisco where this particular strain was isolated. This prokaryote is a bacillus or rod-shaped gram-positive anaerobe that is also facultatively fermentative, which means it can produce a variety of fermentation products, including lactic acid, ethanol, and CO2, without oxygen. It is postulated that the success of L. St. Francis and sourdough starters particularly is due to its preference of fermenting maltose rather than glucose, meaning it does not compete with yeast for the same energy resource in the mixture. To cultivate one of your own, start with equal parts flour, say all-purpose or whole wheat, and room temperature filtered water. Stir until it's thoroughly mixed, then cover it with a tea towel to allow for exchange of air and gases into and out of the vessel. Leave it in a warm environment, ideally 70 degrees Fahrenheit, to let fermentation begin. The next day, remove half the mixture and feed it again with equal parts flour and water. Keep doing this for 1 to 2 weeks until you have something that looks like this. Then store it in the refrigerator in an airtight container, feeding it once a week or as needed. You now have magic in a jar to bake your very own unique sourdough bread, just like the ancients did.
Hi, I'm Nick Nesselrode, and this is Listeria monocytogenes. In the human microbiome, there are 10 times more microbial cells than human cells living in and on us, and thousands of different species throughout the body. Most are commensal or mutualistic, but some can be pathogenic. Many of them aid in digestion, activate and strengthen the immune system, provide antimicrobial protection, maintain the endothelial walls of the gut, and influence metabolic rates. Factors like taking rounds of antibiotics, changing your diet, and even age can significantly affect our gut microbe communities. Recent research has been focused on microbial activity in the gut in relationship to things like clinical obesity, diabetes, Crohn's disease, and allergies. Though microbes live in our mouth, our skin, our nasal cavity, our stomach, our small intestine, most microbes actually live and digest in the colon. Microbes in the colon aid in digesting foods that the human body cannot or inefficiently digests. Within the genus Listeria, these are gram-positive bacillus-shaped firmicutes, and they are 1/2 to 2 micrometers in length. They're facultative anaerobes and can grow at temperatures between 39 degrees and 111 degrees Fahrenheit. There are 21 known species of Listeria, Listeria monocytogenes being the most notable because it causes listeriosis. Other species like Inocua are non-pathogenic, and some just have a limited pathogenicity. Listeria monocytogenes is particularly notable for pregnant people. They are 12 times more likely to get listeriosis, and this is due to reduced T-cell immunity and increased progesterone levels. So oftentimes it often goes undiagnosed because of the flu-like symptoms, but can become really serious and cause things like meningitis and seizures if the bacteria gets into your blood. For prevention of Listeria, the CDC recommends some food safety and food storage measures like cooking your food thoroughly, washing hands and surfaces, and rinsing fruits and vegetables. Listeriosis is typically diagnosed through a blood test and treated with antibiotics like ampicillin and gentamicin. Even though we probably all heard of Listeria, it's actually quite rare, only 4 in 100,000 people. And usually our microbiome, the microbes in our gut, can usually prevent the bacteria from getting into our bloodstream in healthy individuals. So usually Listeria is really only an issue for people who are immunocompromised or for pregnant people. So everybody wash your hands. Thanks.
Hello, for homework assignment number 1, I chose the bacteria strain of Lactobacillus rhamnosus. This is a bacteria strain that has specifically been studied for its effects on infants and the immune system. Looking at the infant microbiome, we now know that the microbiome in infants is shaped by how a baby is born. A baby who is born vaginally will have a microbiome that is built largely of bacteria that they pick up from their mother's vagina and anus. And these are like very important foundational bacteria for the microbiome. And infants who are born via C-section have a microbiome that's dominated by bacteria from the skin and from the hospital environment. And it's now known that the infant microbiome is in flux for the first 3 years of their life, and then usually by the time a child is 3, their microbiome looks a lot more like that of an adult. And the microbiome, while it's developing, can be shaped by the diet, the use of antibiotics if a child is young, is very sick when they're young and on a lot of antibiotics. That changes the shape of their microbiome. Looking at Lactobacillus rhamnosus, it was studied in infants who were exposed to the bacteria in pregnancy. It was given to the mothers in the last trimester of their pregnancy, and then to infants in the first months of their life. And this bacteria was found to modulate immune cells to better withstand allergens that cause things like dermatitis and eczema. And exposure to this bacteria reduced the risk of those immune diseases by up to 50%, which is a pretty remarkable statistic.
Microbes and me. What is a human microbe? A human microbe consists of bacteria, archaea, viruses, and eukaryotes that reside both inside and outside our bodies. They are a collection of organisms that inhabit and interact with humans. 10 to 100 trillion symbiotic microbial cells in each person. We need it to survive and regenerate, and they help with digestion, and the mix of microbes in our guts can help with using and storing energy for food. Some positive and negative outcomes. They help humans with digestion and protect against pathogenic invasions and modulation of the immune system. Some benefits on health that they have is immune health, heart, weight, and other aspects such as the gut microbe. Some negative outcomes is that the gut microbe can contribute to heart disease by producing trimethylene oxide. TMAO is a chemical that contributes to blocked arteries, which may lead to heart attacks and strokes. Another example is the norovirus, which can cause acute gastroenteritis and inflammation of the stomach and intestines. Antibiotics. What makes antibiotics very essential to treat infections is they treat infections such as open wounds like deep cuts to help kill the bacteria and prevent further spreading, even though it can also kill the good bacteria, which causes sickness. Overconsumptions of antibiotic can also cause bad bacteria to multiply. This is why it is very important for physicians to prescribe a certain amount. The human gut microbe. It is a microorganisms that include viruses, bacteria, archaeas, fungi, and protozoa that live in the gastrointestinal tract. The gastrointestinal tract is an environment with varying pH levels, primarily anaerobic conditions and rich nutrients. Its acidic pH supports digestion and pathogen controls. Therefore, the more neutral pH in the small intestines, it supports diverse microbial life. Its gut mucosal barrier protects the intestinal lining and provides habit for beneficial microbes and interaction with the immune system to help maintain balance in the small intestines, which support diverse microbial life. A healthy microbe is a bacteroides. A common virus is a norovirus known as the stomach flu. And initially how it takes place is the innate immune system responds with cytokine release, activating adaptive immune cells like cytotoxics, T cells, and natural killer cells. Viral infections can alter the microbiota composition, potentially leading to thrombosis that weakens the gut barrier and increases further infection. Due to the replication, the virus is shed in feces, allowing for transmission. As for the long-term effects, viral infection can cause persistent changes in the gut microbe, affecting overall health and potentially contributing to other diseases such as hypertension, diabetes, and fatty liver disease. So here I have an example of how a norovirus looks. It is a non-enveloped single-stranded positive sense RNA genomes of approximately 7,500 nucleotides that belong to the Calyciviridae family. It is a cup-shaped morphology around 27 nm in diameter and anaerobic. It includes an overlapping open reading frames, which begins with the first stage, which encodes a polyprotein with structural and non-structural proteins. going to the second, which encodes to the major capsid protein, and the last, which encodes the minor capsid protein. It is the leading cause of viral gastroenteritis, and although it is an unhealthy virus, it can be beneficial by making the gut healthy again through antibiotics. Hello, I'm Emily Bauman, and this is the human microbiome and Streptococcus salivarius. The human microbiome is made up of trillions of microorganisms that live in areas like the gut, skin, mouth, and respiratory tract. These microbes are essential for maintaining overall health by aiding in digestion, supporting the immune system, and producing essential vitamins. In the mouth, the microbiome includes beneficial bacteria like Streptococcus salivarius, which helps maintain oral health. However, factors such as antibiotics can reduce microbial diversity, while diet can either enhance beneficial microbes or foster the growth of harmful ones. Age also influences the microbiome as it continues to evolve over life. When the microbiome becomes imbalanced, it can lead to negative health impacts, including metabolic disorders and autoimmune diseases. In the mouth, this imbalance can result in tooth decay, gum disease, and bad breath. The mouth's environment is shaped by several abiotic factors, such as pH levels ranging from 6.2 to 7.6, temperature around 98.6 degrees Fahrenheit, saliva, which contains enzymes and electrolytes, and the presence of both aerobic and anaerobic environments. Different parts of the mouth have their own microbial communities. Streptococcus mutans is found on the teeth, gingivitis causing bacteria on the gums, and Streptococcus salivarius is common on the tongue. The interactions between these microorganisms and the host are critical, involving mutualism, immune system modulation, and defense against pathogens. An imbalance of microbes in the mouth can lead to tooth decay, gum disease, and bad breath. Streptococcus salivarius belongs to the genus Streptococcus. It is gram-positive and appears as spherical chains. It is a facultative anaerobe that thrives in the oral cavity and upper respiratory tract, with close relatives such as Streptococcus vestibularis and Streptococcus thermophilus. In the mouth, Streptococcus salivarius acts as a probiotic, promoting a balanced oral microbiome, improving mucosal barrier function, and helping to combat infections. Here's my works cited. Thank you.
There are so many microbes that call our bodies home. Most of these microbes are harmless, such as those in our gut, aiding in our digestion. While harmless, any disruption to the naturally occurring quantities of different microorganisms can cause uncomfortable or infectious results. Taking a closer look at places microbes exist in our bodies, we have the spaces of the apocrine sweat glands. The secretion of sweat, as well as the constant friction in these areas, produces a very moist, warm, and nutrient-rich environment that is perfect for microbial growth. Staphylococcus hominis is a species of bacteria that lives within our armpit and groin regions. To break down the name here, Staphylococcus is a gram-positive cocci that forms in clusters. Hominis means human in Latin, so when we put the two together, we get Staphylococcus hominis. This bacteria is responsible for the foul odor some people produce. In other words, when this bacteria feeds on our sweat, sulfurous and onion-like odors are produced. While it's relatively harmless to our overall health, it might not be to our noses. Allowing this bacteria to sit and multiply on the skin will result in more amplified and intense odors. In the super rare case that it does find its way into the bloodstream, that can cause an infection. Typically only happens in hospital settings, specifically when patients are getting some sort of medical device implanted, like a heart valve. Because the infections typically occur in hospital settings, it has evolved to be multi-drug resistant. On the other hand, its ability to form biofilms on our skin leads scientists to believe that it may help protect against pathogens.
Guys, today we're going to be talking about the diversity of the gut microbiota. Starting us out with two really fun facts. One, there is more neurons in your gut than in your spinal cord. And fun fact two, the weight of the human microbiota ranges from 2 to 5 pounds, which is the weight comparable to the human brain. What is our human microbiota? As we found out in lab, microbes are ubiquitous. These guys are living on us, in us, around us. They are our team of helping healthy buddies. It is so important to have this because these guys are what help keep us healthy. They help us digest food, make vitamins, fight off infections and bacteria. We need them. They are our gut superheroes. And why the gut? They like the gut because it is the perfect environment for them. It is cozy, it's the right pH, it's the right temperature, it's the right humidity, there's lots of nutrients, it's a friendly place for them to be, unless we are overusing antibiotics and eating garbage food that has had all of the nutrients taken out of it, including the fiber. Yes, everyone say hello to Faecobacterium praunitii. This guy is in our gut helping by producing butyrate, which feeds our gut cells and keep it healthy, helps keep inflammation away. And if you know anyone with Crohn's disease or IBS, they are going to want more of this healthy microbe. Now, you've heard me say diversity 1 or 2 times, and I wanted to bring it together. Diversity in education, diversity in diet, both of these things create a more healthy environment. And I'm really grateful for PCC and the diversity that I even see within our class that helps bring a more well-rounded experience to all of our education. So in a nutshell, our superstars, one of which, Faecobacterium praunitii, and also our fruits and veggies, our yogurts, are things that help feed this healthy microbiota to keep us healthy and thriving. Thanks, everybody.
The human microbiome, healthy skin, and Staphylococcus. Only relatively recently in the history of medicine, the human body's microbiota, or communities of microorganisms, are of considerable interest in the further study of human health. The skin is often considered an environmental desert when compared to places such as the GI tract. However, comprehensive studies of skin microbiota still reveal diverse niches within the skin biome, such as moist, hairy underarm regions as compared to dry, smooth forearm regions, and microscopic niches exist, such as the structural unit of the hair follicle, which provides a microaerophilic environment. Across the skin microbiome, around 1,000 microbial species are found, most of them bacteria, and 3 of the most commonly found genera being Corynebacterium, Propionibacterium, and Staphylococcus. Skin bacteria species interact competitively with each other, such as in the inhibition of the opportunistically pathogenic species Staph aureus by coagulase-negative Staphylococci species, and bacteria interact with the human host in the range of symbiotic relationships, mutualistic, commensal, and least common parasitic in the case of pathogenic bacteria. An ideal balance of this skin bacterial community does exist and helps maintain human health. Alterations to the community may underlie increases in diseases such as atopic dermatitis. 2 important skin bacteria to focus on further for their relationships to both each other and to the human body are Staph epidermidis, which is a key inhabitant of the healthy skin microbiome and a mutualistically symbiotic organism with humans, and Staph aureus, which is also found on healthy skin but is more virulent as a potentially opportunistic pathogen. Staph epidermidis and Staph aureus both belong to the phylum Bacillota and the genus Staphylococcus and are both gram-positive, catalase-positive, and facultatively anaerobic cocci, while Staph aureus produces enzymes contributing to higher pathogenicity such as coagulase and staphylokinase, and Staph epidermidis is one of the aforementioned coagulase-negative Staphylococci species. These 2 species compete vigorously with each other to maintain colonization on the skin, with implications for health, given that one is more pathogenic than the other and can cause skin infections if it grows unchecked and breaches body barriers. That namely is Staph aureus. To compete, Staph epidermidis carries out activities such as production of a serine protease that disrupts biofilm formation of Staph aureus, and Staph epidermidis produces phenol-soluble modulins that exercise antimicrobial activity against Staph aureus, and even cooperate synergistically with our innate immune system's own antimicrobial peptides.
Hi, I'm Amy Hawkins, and I'm discussing Fusarium and the human intestinal microbiota. So the intestinal microbiota contains a lot of different microorganisms, including fungi, which Fusarium is a part of. The intestinal characteristics include villi, blood and lymph vessels, mucus, digestive fluid. It is pretty moist. It has 3 different zones. Each of those have different microbiota. It's gram-positive in the duodenum, gram-negative in the ileum. And Fusarium is something that you might find in your intestines if you are unfortunate. It is generally pathogenic in plants and animals. Although if you've ever had corn, that meat replacement with the QO, it is a type of Fusarium. Fusarium is an ascomycete. You can see its lineage on this slide. And some of its macroscopic features are in the first picture. It's a white hyaline mold with developing coloration in the center. And it is under the microscope called microconidia, or canoe-shaped. So the mycotoxins that Fusarium produces are actually what cause disease, not the Fusarium itself. These 3 are the most common ones, and you can see their method of action here. Fumonisins are pretty common because they grow on corn, which is how a lot of people get sick, by eating bad corn. Fungus spores in general can affect galactose metabolism, so they can reduce the metabolites involved, cause inflammation, intestinal barrier damage, and increased absorption. Fusarium can also cause superficial infections like nail fungus. Usually that's Fusarium falciform, the specific species. And a more invasive infection would be called fusariosis. That's Fusarium keratoplasticum in most cases, and it can be over 50% fatal. In the USSR in the 40s, there were over 100,000 people that died from alimentary toxic aleukia because of the mycotoxin T2 from Fusarium, and also Akakabe poisoning in Japan and moldy corn disease in the US is from trichothecenes. A lot of this was from eating bad overwintered corn. So don't overwinter your corn. You should be okay.
The adult human body has about 10 to 40 billion cells, and scientists estimate that 10 times the amount of bacteria, fungi, and other microorganisms reside on the body. The microbes make up the microbiota. They provide protection against invading pathogens and therefore play a major role in immune protection. The microbes within the GI tract is called the gut microbiome and is important for overall health because it breaks down essential nutrients, drug metabolism, and immune development. Clostridium difficile can be found in the intestinal tract. In the healthcare field, both the bacteria and the disease are commonly referred to as C. diff. C. diff is a gram-positive anaerobic bacteria with capacity for endospores. Within a healthy gut microbiota, C. diff can be found in small amounts suppressed by more dominant anaerobes which prevent overgrowth or colonization. However, in large amounts, C. diff can cause an infection, which can lead to diarrhea, cramps, and the development of colitis, and potentially severe cases can lead to death. So you might think about C. diff's presence in a healthy gut microbiota as a small gathering, and then C. diff infection as a high school house party. Like in the movie Mean Girls, she thought she was hosting a small gathering, but it turned into a raging party. So in small amounts, it's okay, large amounts, not so much. When there is damage or changes to the gut microbiota, imbalances in the bacteria allow C. diff to take over. This can happen with the use of broad-spectrum antibiotics. C. diff is contagious, and hospital-based infections are common as C. diff is resistant to alcohol-based cleaners. Traditionally considered an infection affecting patients in healthcare facilities, C. diff is now increasingly identified in community among people with no recent hospitalization or stay in healthcare facilities.
Hello, this is Cassandra Carroll, and this is my Microbes and Me Homework 1 project. So what is the human microbiome? The human microbiome consists of all the microbes that exist on and within us. It is tailored to the individual, and as in a healthy individual acts as a line of defense against pathogenic organisms. That being said, any disruption of that microbiota, such as with the use of broad spectrum antibiotics, can have negative effects on our health. The skin microbiome, the skin is the largest organism of the human body. There are very multiple environments on the skin that give rise to different microorganisms. So the moist regions, we have Staphylococcus and Corynebacterium primarily. In sebaceous regions, we have Cutibacterium. And in dry regions, which is actually the most diverse region of the skin, has Actinobacteria, Proteobacteria, Firmicutes, and Bacteroidetes. Interestingly, with the skin, interactions with the environment and the skin is actually essential to the development of the immune system. One of the microorganisms I focused on was Cutibacterium acnes, which is the causative agent of acne. It is gram-positive and lipophilic in nature. It is an aerotolerant anaerobe, so it can survive brief periods of time in oxygenated environments, but primarily enjoys anaerobic environments. It normally lives on healthy skin, but when we have overactive sebaceous glands, usually due to hormonal reasons, it creates areas of hypoxia on the skin, and because it is an anaerobe, leads to excessive growth of C. acnes, which causes that classic inflammation, which results in the acne appearance.